It is my distinct pleasure to introduce Elena Suglia, a graduate student in Pop Bio, for her exit seminar today. I will admit that I really struggled with what to highlight from Elena's professional and academic record because she's been busy. And her academic and her activities and accomplishments are so multifaceted, but I will try, and I have notes to help. So Elena got her bachelor's degree in biology from Brown University, where she got involved in research, science communication, and mentoring and social justice right away. And this is a hallmark of her career to date and what she hopes to continue in her career moving forward. At Brown, Elena conducted an honors thesis on trophic dynamics and salt marsh die-off and co-authored a paper in Ecology Letters on that topic. Not only that, she also wrote and illustrated a children's book on anthropogenic threats to salt marshes, along with other science communication articles and briefs and activities. At Brown, she was also deeply involved in teaching and mentoring her peers in the classroom, at science communication workshops, and in the field, as well as leading social justice efforts. Elena then worked in the Winfrey Lab at Rutgers on projects relating to restoration of pollinator habitat for native bees, before we managed to get her to come to UC Davis as a research technician for Annie Schmidt, working on collaborative research between our groups. That was when Elena applied for the NSF Graduate Research Fellowship and was awarded it, and joined my lab in 2017, where she dove into research on a cool, very complicated native California wildflower that she'll be telling you about today. For her research, Elena tackled intensive fieldwork, complex common garden experiments, and population genetic studies and analyses. And I won't steal her thunder and tell you more about that research. I'll let her tell you about that work. But from here, Elena will go on to be a Sea Grant Fellow, where she'll be working with the State Water Resources Board. That starts in February. There were a couple things that I wanted to highlight about Elena and her time here at UC Davis and in Pop Bio. First, that she's been dedicated to and very effective at mentorship, science communication, and diversity, equity, inclusion, and justice from her very first days here. For Elena, these components are deeply integrated into what it means for her to be a scientist, professional, and a citizen. She has a long list of service and accomplishment in these areas, and she has made a tremendous impact from my lab to the grad group, to the Eve department, to across campus and local communities. A few of the highlights include receiving the first Sharon Gray Memorial Award for mentorship, her mentorship of K-12 students at local underserved schools and through national programs, publishing several SciComm articles, co-founding the Evolution and Ecology Grad Preview Program, her work on the Diversity Committee for Pop Bio, and her work as the graduate advisor to the Dean of CPS on diversity, equity, inclusion, and justice. And that's just a few. There are just way too many accomplishments to share them all. The second thing that I want to highlight about Elena is just her drive and ability to develop and support community. She has been a source of cohesion in my lab and in Pop Bio and beyond. I'm her official advisor, but there's many people in this room that have also served as mentors and advisors, notably Annie Schmidt, who is, I would say, an unofficial co-advisor and important mentor for Elena, as well as Sharon Strauss, who I think is probably on Zoom. And despite my cautions, an example of this is despite my cautions about how difficult it would be to schedule committee meetings, Elena has 5 members of her dissertation committee whom she's been very good at keeping in the loop and involved in her research, and has been very supportive of those activities and research, and I'm deeply grateful to them as well. But I think this just illustrates how collaborative, engaged, and effective she is at building connections and community. And this community is proud of her, and will miss her when she moves on. Okay, I'll give her some time to tell you about her science. Nowhere. You have this worked out. This is my fault for switching on here.
I think it's on extend. I think it peaked out on the left a little.
Yeah, I think it.
Did you lose your mouse too?
Okay, hold on.
Oh, there we go.
Okay, we just need to share it with the
Yeah.
online crowd.
Almost there.
It's, yeah.
No, it's this one. Yay.
And
All right. Thank you, Jenny, for that wonderful introduction. So today I'm going to talk to you about my dissertation research on divergent phenological and fitness responses to precipitation and herbivory across an elevation gradient in a native California wildflower. So in a variable and changing world, timing life events to coincide with favorable abiotic and biotic conditions is critical to individual fitness, population viability, and species persistence. For example, a favorable biotic condition could mean timing plant reproduction with pollinator availability, or sessile organisms like plants which can't move to avoid herbivores may instead time growth so that herbivores are least abundant at that time. Timing is also really important in seasonal environments because bad timing could leave you like this plant trying to grow in the snow. And so because of this, selection favors individuals that time events in a way that maximizes fitness. Therefore, many organisms evolve responses to environmental cues like moisture, temperature, and day length to time events like emergence, growth, and reproduction to maximize their fitness. For example, plants often germinate after it's rained and flower after they've accumulated enough heat and light. Timing of flowering is an especially critical phenological event in terms of fitness consequences. So flowering too early can incur fitness costs by exposing buds or flowers to frost, reducing fecundity. On the other hand, flowering too late can expose plants to late season drought or the onset of winter conditions. Of course, the best time to flower will depend on the environment that the plant experiences, which is why life history responses vary with environmental conditions. So say you're this high elevation plant living at the top of a mountain where you flower in spring after the snow melts and the growing season ends whenever you run out of water. So in a wet year, maybe there's summer rains or a lot of snow melt, you might want to grow for longer to maximize resources for reproduction and flower later at a larger size. And in a dry year, you might instead flower earlier at a smaller size to complete reproduction more quickly. Say you're the same plant, but now you've been browsed by a mammalian herbivore, like this rabbit. This could really slow down your reproduction for the year. You've lost all this biomass and you're half the height you were before you got chomped. So if you're a plant that lives for only 1 year or an annual, that means you need to flower within one growing season. Although maybe at a smaller size than you would have liked before you got eaten by this guy. So in a wet year, if annual plants want to flower, they might actually have enough time to reaccumulate biomass and compensate for that herbivory by not suffering any fitness losses. But if it's a dry year, they may actually hurry up and flower really quickly before they run out of water. And they could then experience fitness losses because they don't have the time or resources to create lots of flowers and fruits. So in this way, herbivory and water availability can interact to affect phenology and fitness responses. But what if in the same scenario, you're now a perennial plant? So you can live for multiple years before flowering.
In a wet year, maybe you go ahead and flower because you have plenty of resources and time to do so, even if you've been eaten. But in a dry year, when the growing season is short, maybe you decide not to flower at all and just recover what biomass you lost and hunker down to wait until the next year to reproduce. So your overall life history strategy can also impact reproductive decisions, like if you're an annual or perennial. So we just talked through an example of how a plant living in the same location can time flowering based on seasonal conditions from year to year, but this same trade-off may occur across locations as well. So in this example, at higher latitudes or elevations, plants may flower earlier at a smaller size than those at low elevations or latitudes. These patterns in flowering time may reflect local adaptation to factors that vary by elevation or latitude, such as growing season length. However, evolutionary adaptation to local conditions takes a lot of time, and with climate change, organisms are being exposed to novel conditions at an unprecedented rate, which may disrupt their adaptation. So the question becomes, what are the biological consequences of these changes? To answer this question, I focus primarily on phenological and fitness responses. So one of the consequences of climate change is altered timing of abiotic events like temperature that organisms historically use to time life history events. Springs are coming earlier and they're warmer, and as these environmental cues change, plants are shifting their phenologies in response by both plasticity and evolution, typically towards earlier flowering. And this observation is consistent with studies showing that selection tends to favor earlier flowering, especially in temperate environments. But shifts to earlier flowering are not always universal, and populations and species vary in the direction and magnitude of responses. So for example, fall flowering species that use photoperiod as a cue may not shift their flowering as much as a spring flowering species that uses temperature. Climate change can also disrupt adaptation. For example, as conditions warm, plants are adapted to conditions that historically existed in their environment several generations ago. So we're seeing a bunch of these examples of local maladaptation and adaptational lag with climate change, or in other words, organisms aren't always keeping up with the pace of climate change. So these are some of the potential consequences of climate change. And we've already seen in nature, there are phenological changes occurring, disrupted adaptation, which can result in negative fitness consequences. So in the face of this change, will organisms be able to persist? We can approach this question in many different ways, but in my dissertation, I investigate plant traits and phenological and fitness responses to different abiotic and biotic conditions along elevation gradients. And specifically, I ask, what are the effects of snowmelt timing on phenological and fitness responses across elevations? And how do elevation or climate of origin interact with drought and herbivory to affect phenology and fitness? And I do so in a native California wildflower, Streptanthus tortuosus, which Jenny mentioned, is a very complicated study system. You'll see why. So this is what the plant looks like across the different environments and life stages. It's a native wide-ranging forb in the mustard family that inhabits dry, rocky slopes and outcrops from Southern California to Southern Oregon. It's a self-compatible species, but sets few seeds in the absence of pollinators and depends primarily on outcrossing for reproduction. So timing flowering with pollinator availability is really important for the species. It occupies a Mediterranean climate characterized by high inter and intra-annual variation in temperature and precipitation across its range. And they tend to be discontinuously distributed across the range and located across a broad elevational climb from 200 to 4,100 meters. So here are the 20 populations I studied in my whole dissertation colored by elevation. And I'm focusing on 2 projects today that each looked at a subset of these populations. So here I mapped the populations onto their climate space and did a principal components analysis, which includes maximum temperature, total precipitation, snowpack, soil water storage, and climatic water deficit, which is a metric of droughtiness from 1990 to 2020. And this first horizontal PC axis describes variation from low elevation with warm, long growing seasons over to higher elevations with short, cool growing seasons. And this second vertical PC axis describes variation along a moisture gradient from wet to dry. So just to give you a teaser from my first chapter, which I won't dive into today, I looked at population genetics and morphological trait divergence across climatic space. And for this talk, I'll just say that I found that at lower elevations, there tend to be more annuals or plants that live for 1 year. And those plants tend to be taller with thinner leaves and fewer branches. And then you see the opposite at higher elevations, where there tend to be more biennials that live for 2 years, that are shorter with thicker leaves and more branches. And occasionally at high elevations, you also see plants that live for more than 2 years and reproduce more than once, or Iteroparis perennials. And we also found some evidence that these patterns are adaptive, which is interesting because they match this fast-slow continuum in both life history strategy and morphology across an elevation gradient. So that's a bit about the populations. Going back to our questions, let's dive into the first project on the effects of snowmelt timing on phenology and fitness. So warming temperatures are causing a decrease in snowpack and an acceleration in the onset of snowmelt in Western North American alpine regions. In this graph, so it shows how in California, the snow water equivalent or fraction of water that the snow holds has been decreasing and is expected to continue to decrease across different emission scenarios. Snowpack in particular is an important cue on mountains. So in mountainous regions, snowpack levels and timing of snowmelt are major structuring components of the growing season, and they dictate the initiation of development as well as the moisture, thermal, and photoperiod regimes a plant experiences during their growth and reproduction. And so in that way, snowmelt can influence phenology and fitness quite heavily in alpine areas. So in these alpine areas, plants often flower right after the snow melts to maximize time for reproduction during the short growing season. So you might see flowering timing tracking snowmelt timing pretty carefully. But when snowpack is reduced and snowmelt is really early in the spring, you can sometimes see this type of nonlinear response where flowering timing is attenuated, as you can see by the shallower slope at very early snowmelt times, which might actually indicate that these populations are approaching a limit of phenological change. Changes in snowpack and flowering timing also affect fitness. So you might see a pattern of lower fitness with earlier snowmelt and earlier flowering. For example, due to something like frost exposure. And this is something that we expect to see more often with climate change. I just put the direction that we expect to see on the x-axis. On the other hand, plants might have lower fitness with later snowmelt and later flowering because they're still trying to complete reproduction during the onset of late season drought or winter conditions like snow or frost. So the specific questions for this chapter: how does flowering timing vary in relation to snowmelt timing? And what are the consequences of snowmelt timing and phenology for fitness? And we're also interested in seeing how these relationships vary across years. And to answer these questions, I conducted an observational field study at Lassen Volcanic National Park from 2018 to 2020, investigating the effects of snowpack on phenology and fitness. And my field sites include 4 populations in Lassen along this elevation gradient. So plants at Lassen are biennials or sometimes idiroparous perennials that germinate in the spring with snowmelt, develop into vegetative rosettes during their first growing season, overwinter under the snow, and then reproduce after emerging from snow in their second spring. So this is the vegetative rosette stage. These are some buds, and this is what they look like when they're flowering. And so to quantify the effects of temperature and precipitation regime on Streptanthus tortuosus phenology, I measured temperature and length of the snow-free period across plots. Here's one of the plots, so you can see that snow is pretty patchily distributed. And we use these eye buttons, which are small temperature sensors that you bury a few centimeters under the soil, and they record temperature, which you can use to tell when the snow melt happened. To quantify phenology, I recorded numbers of flowers and fruits every 7 to 10 days throughout the growing season on a maximum of 100 individually marked plants at each of those 4 sites. Here is a plant with a little bird band on it that I used to identify the individuals. And I quantified fitness by tracking survival and measuring fecundity by counting the maximum number of fruits each individual produced. So there's a lot of variation in temperature and snowmelt timing in this study system, which makes it an interesting place to do this study. So you might expect that with increasing elevation, you would see the snowmelt day of year correlate linearly. But instead, at Lassen, we see this interesting curve where the higher elevation populations here in blue are actually melting out from under the snow earlier than you'd expect, given how high up they are in elevation. We aren't quite sure why this is the case, but this population is on the Lassen Peak Trail. Maybe some of you have been there. It's a pretty exposed south-facing site, and it gets a lot of direct sunlight. It's also on a volcano, so there might be some geologic activity that might make it warmer than usual, but I'm not a geologist, so I'm not sure. But it provides a unique opportunity to decouple elevation, snowmelt, and temperature in this system. So for our hypotheses, these were the 2 scenarios that we talked about potentially occurring. And I predicted phenology would track snowmelt timing pretty closely with individuals experiencing early snowmelt flower. Individuals that experience early snowmelt would flower earlier than those that experience late snowmelt. I also expected that individuals in late snowmelt patches that flower later in the season would have lower fitness because at Lassen, the growing seasons are really restricted. There's so much snow that there's only a few months for the plants to do their entire life cycle. And then at the end of the season, you get a lot of frost and snow coming down really quickly. So this is actually a picture of a Lassen plant that flowered a little too late. And all of these little brown pods are supposed to be their fruits, but they've just been exposed to frost and had like 100% fitness loss. So that's what I expect to see for fitness trends. Okay, diving right into some of our results. So here you're going to see snowmelt day of year on the x-axis and peak flowering day of year on the y-axis, which we defined as the day on which each individual had the highest number of open flowers that we counted at those every 7 to 10 day intervals. So here, each point is an individual colored by elevation, and lines are indicating the year. And overall, we see earlier flowering with earlier snowmelt, but our responses were actually attenuated at the earliest snowmelt times, as you can see by this reduced slope at really early snowmelt day of year. And as I said, some other studies have found these results, and they think that this type of nonlinearity may indicate populations are reaching a limit to their plastic phenological response when snowmelt is really, really early. Then looking at fitness, we have snowmelt day of year on the X, and then we looked at 2 metrics for fitness. First, probability of making fruits given that you flowered, and then your fecundity, which is the maximum number of fruits for each individual. So probability of making fruits was lower with later snowmelt day of year. Across all years, that's what we expected to see. But then fecundity was a bit lower in 2018 and 2019, but then higher with later snowmelt in 2020, just slightly. We also looked at relationships between flowering timing and fitness. So we have our peak flowering day of year on the X, and then the same two fitness metrics on the Y. And again, we see differences in patterns across years. So in 2018 and 2019, we see what we expected with lower fitness with later peak flowering. And then 2020, we have the opposite pattern. And then with our fecundity results, we actually saw a slight increase and a non-linear trend across all years. So kind of interesting. So why do we see different fitness responses to snowmelt and flowering timing across the different years? This might be because in 2018 and 2019, we see our expected pattern, but then in 2020, it's actually the warmest, wettest year, which might have extended the growing season length. So we expected higher fitness with later snowmelt because the snow-free period is really short, and that's how we define the growing season length, the snow-free period. But moisture and temperature also affect the growing season length functionally for the plants. So if 2020 was especially warm and wet, then flowering later may have allowed the plants to take advantage of those better conditions to maximize fitness. We just didn't incorporate that into our definition for growing season length. So to sum up what we found for this first project, we saw that flowering time advances with early snowmelt, but the non-linear trend we observed with a shallower slope at the earliest snowmelt times might suggest that Streptanthus tortuosus is approaching a threshold in climate sensitivity, which might limit its ability to respond with even earlier snowmelt that's projected with climate change. We also found that fitness responses vary by year. So generally, early snowmelt conferred fitness benefits, except when the growing season was lengthened. Going into our second chapter. So climate change is shifting plant herbivore interactions, and consistent with paleontological records showing higher rates of herbivory during warmer periods, a recent meta-analysis found that warmer temperatures and drought increases florivory. And we've also seen animals, including lots of herbivores, migrating upwards in elevation and latitude to keep pace with climate change. So both of these trends could lead to plants being exposed to novel herbivores or higher rates of herbivory than they experienced in the past. So this meta-analysis shows the effect sizes of herbivore-induced damage to climate change manipulations. And so you can see that treatments such as drought and temperature increase by the positive effect size, herbivore damage on flower and leaf tissue. And populations from areas that experience high herbivory pressure historically, such as low elevation, warmer environments, often evolve ways to deal with herbivory, like chemical or structural defenses, and they may not actually experience very large fitness losses when exposed to herbivory in something like a common garden experiment where you have a control treatment and an herbivory treatment. But if you took plants that evolved in areas that had historically low herbivory pressure, like higher elevations, they actually might experience greater reduction in fitness compared to lower elevations. So populations might be locally adapted to herbivory pressure along environmental gradients, like elevation or latitude. So to answer this question, I use these 8 populations across latitudinal and elevation gradient, and to characterize herbivory pressure at these populations in the field, I collected data on the presence or absence of herbivory on 60 individuals at each population, and defined herbivory pressure as the proportion of the individuals that experience herbivory. And as you can see here, populations at higher elevations experience lower herbivory pressure than those at low elevations, which is what we expect to see. Individuals also vary in the amount and type of herbivory they experience in the field. So these are Strathanthus tortuosus individuals across life stages. You can see this vegetative individual has a lot of insect chewing on it. This one, which would have been a flowering individual, got eaten down by some mammalian herbivore. And this plant trying to make fruits has been eaten on pretty much every organ, the stem, the leaves, fruits. Looks like grasshopper damage to me. And here are some of the types of mammalian herbivores that we see in the field. The American pika is our cutest herbivore at high elevation sites. And like you can see it's doing here, it removes large amounts of tissue from Strathanthus to store for the winter. You could also have rabbits and deer, but they aren't as cute. So this is the same PCA you saw before, except now I've shaded out the populations I'm not using in this study. And as a reminder, climate space for these populations primarily varies along the elevation gradient from low elevations with warm, long growing seasons to high elevations with short, cool growing seasons. So I conducted a common garden experiment exposing plants to varying simulated herbivory and water availability. We had 2 herbivory treatments, a control, no clipping, and then our herbivory treatment where we clip plants during flowering. And we observed that plants experience the most damage at this phenophase in the field. We also clip plants in a way that mimics mammalian browsing that you'd see in the field, kind of like the pika. And then we assigned plants to one of 5 watering treatments. And we did so across the range of water conditions experienced by our populations in the field. So these points are precipitation per population during the 8-month-long water year from October to May, on average since 1990. And the populations are shown in order of increasing elevation. And then these horizontal lines represent the water availability levels we use to create our watering treatments, which we translated into millimeters per day for the purposes of dispensing water to the plants. So you can see here, the lowest watering treatment is 40% lower than the driest site. And the highest is this well water treatment for comparison that was 60% wetter than the wettest site. So then we crossed our 2 herbivory treatments with our 5 watering levels and randomly assigned 8 plants per population to each treatment. And then we collected flowering phenology and fecundity data weekly on plants, which were placed in trays outside on benches. And shout out to Danielle, who was my partner in crime for this experiment. Thank you for all the help. And then we had a catastrophic drought in the middle of our drought experiment. So the watering software malfunctioned and stopped watering the plants completely during a heat wave. So there was a lot of mortality. And after this, we just felt like we couldn't really trust the data that happened after the accidental drought. So we essentially ended the experiment and used the data we collected prior to the drought event. So just to briefly take you through this timeline, we took the plants outside, gave them all the same amount of water at first, and then we acclimated them to their different 5 watering treatments. As the plants started flowering, we gave them their herbivory treatment, and then measured fecundity a few weeks later, right before the drought ended the experiment. Okay, so for our hypotheses, we predicted that drought and herbivory would interact. So here, if you had water on the x-axis and fitness on the y, we expected higher fitness with more water in both the control and herbivory treatments, but more intense negative effects of low water in the herbivory treatment. We also expected that plants would maximize performance under conditions that are more similar to their environment of origin. So here, if we had home herbivory pressure on the x-axis, fitness on the y, and just showed 3 different watering treatments, low, medium, and high, we predicted that when plants were clipped, plants from environments that experience higher herbivory pressure would outperform the plants that were from areas with historically low herbivory pressure. So we see the slopes are always positive, but the benefit of being from a high herbivory pressure area is increased with more water. So here we have some results, just like the graph you just saw. It's a little complicated, but we have herbivory pressure on the x-axis here, and this actually correlates really highly with elevation and growing season length. So just to remind you here, I've shown that at high elevations, we have low herbivory pressure and short growing seasons, and the opposite at low elevation. This left panel is for the control treatment, the right is for herbivory, and we have probability of reproducing here, which is the same as for the last chapter. It's the probability of making fruits given that you flowered. So in the control treatment, we see pretty much all the plants are able to make fruits, no matter what watering treatment they're in. But then in the herbivory treatment, things get pretty interesting. So plants from environments with higher herbivory pressure at home and longer growing seasons at lower elevations tolerate herbivory better. So for 4 out of 5 of these lines, the slopes are increasing the way we expected. But at the lowest water level, we see the opposite. So these results are largely consistent with local adaptation to herbivory pressure along an elevation gradient. But growing season length, as we said, also varies along elevations. So you might expect that these high elevation populations would respond to herbivory by rapidly growing the reproductive structures so they can finish flowering in the brief growing season. The reason we saw the opposite, so high elevation populations with shorter growing seasons were generally less likely to attempt reproduction following clipping, might be because individuals can display this iteroparis perennial life history strategy. So after being herbivorized, they might not have enough time left in the growing season to flower and make fruits again. So instead they might just hunker down, wait to like get through the winter, start reproducing again the next year. That might be why that's happening. Then looking at fecundity against experimental watering treatment. Here we have our populations as panels in increasing elevation, increasing order of elevation. And we'll have herbivory treatment again with control as green, pinkish as herbivory. So drought and herbivory both did decrease fecundity as we expected for the most part, except in some high elevation populations where fecundity was lower with higher water only in the herbivory treatment. So this is interesting because if we think plants are using water as a cue for time remaining in the growing season, we would expect them to have higher fecundity with more water, so it could indicate to them that they have more time and resources to continue reproducing. So the fact that we see the opposite for some of these populations might indicate that individuals at high elevations are using cues other than or in addition to water to make life history decisions, like whether to try to make fruits. So in summary for this chapter, plants from environments with higher herbivory pressure and longer growing seasons at low elevations tolerate herbivory better. It's largely what we expected, except at the lowest water level. And herbivory and drought generally depress fecundity as we expected, except for some high elevation populations when experiencing herbivory. So under climate change, we expect to see decreased snowpack, increased aridity, and altered herbivory regimes. So what can our conclusions tell us about how the species may respond to these changes and biological responses to climate change in general? Back to our questions for the first chapter. We see that phenology tracks snowmelt timing, but might be approaching plastic limits as indicated by the nonlinear trend we observed. So plastic phenological responses to snowmelt could buffer plants from some of the negative impacts of climate change, especially maybe in the short term. But as climate change continues and snowmelt becomes even earlier, they might reach a tipping point in their ability to respond. We also saw that fitness is maximized at earlier snowmelt times unless growing season is lengthened by good conditions. So this is interesting because there are a number of studies finding plants flowering earlier with earlier snowmelts and experience increased frost exposure, which reduces fitness. But at Lassen, as I said, snowpack is so high that tipping points related to snowpack might actually be a ways off in the future. So for example, even at the earliest snowmelt times, the temperatures are still above freezing when the plants emerge, so they aren't going to be exposed to frost. But these other study systems showing the opposite result might be a harbinger of what's to come for Lassen. So plastic responses and higher fitness at early snowmelt might provide short-term benefits to plants facing climate change, but not necessarily in the long term as conditions continue to change. So for the second chapter, we saw that individuals from lower elevations generally display higher herbivory tolerance, especially when water availability is high. This is consistent with local adaptation along an elevation gradient, but these patterns are potentially explained by herbivory pressure, growing season length, or something else that varies along elevation. So local adaptation to low herbivory pressure may disadvantage these high elevation populations if herbivory increases with warming or if animals migrate upwards in elevation to track their optimal niches. At the same time, the ability of low elevation populations to plastically maximize fitness, responses to herbivory when water is available, may be advantageous for those. So overall, changes in drought and herbivory pressure expected with climate change are likely to pose significant challenges to Streptanthus tortuosus populations at high elevation specifically. Okay, so to conclude, we saw that Streptanthus tortuosus populations display variation in phenological and fitness responses to precipitation and herbivory across an elevation gradient. Shifts to earlier snowmelt may not negatively impact fitness in the short term, but the long term might be a different story. And low elevation populations may overall be better equipped to cope with herbivory and drought. I have all my acknowledgements. So thank you to Jenny, my advisor. It's been there through all the ups and downs, which there have been many. And the Bremer, Strauss, and Schmidt labs have all been this really integral group and community that has supported everybody connected with them along the way. I have my QE and dissertation committee members up here, Annie and Sharon, as I said, super integral. We also had Rachel Bay, Louis Yang, Elizabeth, and Andrew Latimer. I wanted to shout out Alec Kiono, who was the best partner in crime when I was a tech in Annie's lab and then started in Jenny's lab. All my fieldwork helpers, too many to put on here, and the Eve office staff. So especially Sherry Mann, who's the best coordinator you could possibly dream of. These are the funding agencies that supported my work. I have a couple photos of the grad group for time. And then my lab community. So Jenny, Annie, and Sharon. Shout out to Danielle for being an awesome collaborator. We've had a lot of really good fun times as a lab over the past 7 years. Here's Alec. This is one of my mentees, Lahari. My cohort, and we all look very little in some of these photos. Y'all were amazing colleagues to have throughout this whole journey. Friends and family, You see a lot of familiar faces from the CPB community in my wedding party. You all have really just been there for me no matter what. I want to especially shout out Hannah Higuera and Alexis Robert Hutchis. Yeah, you guys are just the best. I couldn't have gotten through this without you. And Hannah, you've been my rock for the past couple of years. My family. So, oh gosh, you guys should get a medal for being here for the past 7 years while I was completing this. Love you all very much. And Colin, my husband, likewise, he should get an honorary doctorate from what he's had to deal with in the past PhD, and I couldn't ask for a better partner. That I'll take questions.
We are getting close to 5, so feel free to head out if you want, but let's go ahead and if anybody has questions, feel free to fire away.
So did you analyze what happens to when all those plants died? Your unexpected drought. Did you analyze whether there were any predictors of who lived, who died?
Yes, we did. It would be great if I had a backup slide for that. We did see some patterns. They weren't related to the herbivory or drought treatments, but they were related to the environments the plant came from. I can't remember specifically which of those factors. A lot of the climate factors are really correlated, so it can be difficult to disentangle them. But I do think at least something related to their climatic water deficit of origin is important. So we can talk later. I'll leave you on a cliffhanger before.
There's one from online. So early snow melt could be at either lower or higher elevation. Did these two areas differ in flowering patterns, and did the low and high elevation sites differ in effects of flowering time on fitness?
So the way that we define low elevations is that they basically don't have snow. And so, yeah, we don't look at that for those populations. The second question.
Did the low and high elevation sites differ in effects of flowering time on fitness?
That might be
The
the
short-
lesson
yeah.
they're talking about.
Hmm?
Maybe they're asking about the lesson study.
Oh, yeah. Okay, so differences across those elevations in that study, I see. Okay, I'm getting all my chapters mixed up now. Because we have my third one, which also was this. So I think the short answer is no. Yeah. The patterns that we saw were robust across elevations.
Great talk, by the way, and congrats. I was just wondering, for your decision to simulate mammalian herbivory pressure, what was that choice compared to like insect or?
Yeah, a couple reasons. So first of all, like I kind of based my entire dissertation around going out in the field and seeing these plants and realizing that phenology was affected most by the snowpack at Lassen. And then I just was hanging out at one site in particular at Lassen where the picas were like living 10 feet away in a burrow, and they just like destroyed all the plants. And it was really interesting to me to see, like, we did find that plants that were trying to flower in one year would, like the ones with just the little stumps that I showed a picture of, they're not gonna flower, you know, after that in the same growing season. So they just revert to vegetative growth, come out the next season. So that kind of inspired both of those studies. The other thing is that when you're trying to simulate herbivory by an insect, there's all these complicated factors that come into play, like the saliva and, you know, how exactly are you clipping them and which insects are you simulating. Here, you're literally taking scissors and flipping the top of the plant off, and you're talking about biomass loss and decisions about allocation. And so it really simplifies all of those things.
Awesome, thank you.
Any other questions? Okay, let's thank Elena.